Hi guys! Welcome to Science Tech for Fun. In this video, I will be talking about A-Level Biology Practical number 6, antiseptic techniques to investigate the effect of antibiotic substances on the bacterial growth. Okay? For this, that's the list of materials that we'll need. So for this practical, we will use paper, Bunsen burner, gloves, masking tape, heat-proof mat, a lightener, or can be a box of matches. I need ethanol 70%. I need antibiotic multi-discs ring, so I will talk about this later. I need my spreader. We can use two kinds of spreader. We can use a glass spreader where we can also disinfect every single time I use, or I can use just a disposable one that after I use, I just dispose. I have my Agar plate that was done in advance. You can check in the previous video how to do it. I have here my bacteria. In this practical, we're gonna be using E. coli, a Pasteur pipette, a A3 plastic sheet with to work on the top. So this is my working station, and Vircon. So let's start. So the first thing I need to do is look after myself. So I need my PPE. What's the PPE in this practical? Gloves to avoid contamination and specs. Why the specs are necessary? Two things. I will be dealing with flame, so something can go wrong every time I'm dealing with flame. And second, because I'm using bacteria, even if the bacteria I'm using in small amounts, in small population, not cause any harm for me, if it's in a big concentration, if it goes in my eyes, can cause eyes infection. So you need to be very careful, okay? So this is not too much, this is for your own protection, okay? First step is disinfect the area. So where I'm gonna be working, my workstation, I need to spray with Vircon and allow it to set for about 1 minute. Okay? After 1 minute, you can wipe it out, and then you take your A3 station. So for this video, I will just wipe out now, but remember, you need to wait at least 1 minute to wipe this out. So now I can take my workstation here, and I will spray as well. Same procedure, okay? I will leave it settle for 1 minute or 2 minutes to guarantee the Vircon will work, okay? So I leave this about 1-2 minutes, and I will do the same. I will dry this out, well dry, and I will put my heat-proof mat. Heat-proof mat have two areas. One is completely smooth, the other one is not, is hard surface. So the hard surface will be facing down, and this area will be facing up. I will spray again. Leave also set for about 1 minute, and I will dry this out. My Bunsen burner don't need to be disinfected because the Bunsen burner will be, in terms of sterile, the sterile part is possible here. Okay, so the Bunsen burner don't need to do anything. So I will light the Bunsen burner, and I will choose the yellow flame. The yellow flame is important when I'm working in this stage to know the flame is on. Okay, so the area where I should work is about 10 centimeters radial from the Bunsen burner flame. Okay, so it's about this radio here. I call this area stereo. If you don't feel confident enough to work with just 10 centimeters, you can work with other Bunsen burner on the side, so you're gonna have 2 stereo areas added. So you need one here, so we have a big area to work in the middle, okay? But one give you about 10 centimeters radio of sterility, okay? After this is all disinfected, I can start to inoculate my bacteria. So now we will go for the inoculation part, okay? So for this demonstration, I will go for the most complicated part, and then the simple ones I don't need to show because it will be exactly the same procedure. The most complicated part would be using my glass spreader. Why? Because the glass spreader needs to be disinfected every time I use it. So this is sterile. This is completely sterile. Can you see the bag is sealed? So when I open this side here, I can remove, taking the handle from the side, and I can guarantee this will be completely sterile. And after using, I will just dispose. So I don't need to worry to disinfect this. So depending where you are, or you might use this, or you might use those, okay? So I will show you this, that's the more complicated or more tricky part, but the procedure for that is exactly the same. Just open, use, dispose. This one have a little more input, okay? So the first thing is I'm gonna leave this in here in the ethanol 70%. The ethanol 70% is concentration enough to kill the bacteria, okay? And I have my Petri disk here and my broth. So the first thing I need to do is put my disc with the agar facing down, okay, in the bottom, and the lid facing up, right? So I can just open up and close this up. The tip of my pipette is contaminated, so I need to disinfect. How I will disinfect it? With my flame, okay? So for this moment, I will change my flame for blue, or at least slightly blue. Why? Because the yellow flame is incomplete combustion. The incomplete combustion will have some particles that will stick here and might affect the bacterial growth, okay? So I need a completely blue flame, and I will come here in the flame and I will place the tip inside, okay? I don't need to heat too much, okay? I just want to disinfect this. So as soon as it's finished, I can keep this close enough here to cool down, and then I will open my bottle, close here, and I will collect my bacteria. After it's collected, I can just close my bottle again, and then I will open my Petri dish, and I'm gonna deposit 2 or 3 drops in the middle, and then I will spread with my spreader glass. So I'm gonna open this up, 1, 2 bottles, I will close, and I will put here. I'm not gonna suck the water, I mean the ethanol, okay? I'm just gonna keep in there. Then I will take this off. Remember, this is ethanol. Ethanol is highly flammable, so you need to be very careful what you're doing. So you're gonna dry this out, okay, to remove the excess of ethanol you might have, and then you're gonna flame it to guarantee this is completely sterile. Then take from the fire, change the fire for slow and wait a little bit. Then you open and you spread your sample in the gel. Okay? How are you gonna do it? I'm gonna show something that's not right, but just for demonstration. So that's the movement you're gonna do. Spread everywhere. Okay? Spread everywhere. But when you're doing it, it's better if you do it here, with the cover in the top, facing you. Why facing you? Because you might cough, you might sneeze, someone can talk, and pathogenic bacteria can go there and will grow pathogenic bacteria rather than the bacteria I want. So this is like a shield. So the correct way is I'm gonna use here and spread everywhere. Okay? Then I close and put it. Just to highlight one thing, it's very important for you to wait the equipment to cool down. Otherwise, you are gonna kill the bacteria. Okay? So for this second part, I will have my bacteria are already inoculated, and then I will use my antibiotic discs. I will take one here, and I will show you how this looks like. Okay, so this is antibiotic disc. So each one of these parts outside is one kind of antibiotic. We're testing what antibiotic might work with this E. coli and which one not gonna work. Okay, bacteria, they are not sensitive to all kinds of antibiotics. Some antibiotics work better with a specific species of bacteria. Other antibiotics work better for other species of bacteria. Okay, that's why when you go in your GP, the GP not gonna give the same antibiotic depending on what you have. Ideally, the GP would know what infection you have to treat you properly. Okay, so here is how we're gonna do. So I'm gonna place this back here, and I will show you how to inoculate. Okay, because I was not putting anything in the bacteria, it's okay, right? So now I will place this in the ethanol. I will remove the excess of ethanol, and I will change my flame again. I'll change and flame this out. Okay, just to kill. Then go for orange again and keep to cool down. Then I will open the lid. At the same time, I will place my antibiotic disc facing the names up in the agar gel. Okay, so I will open this up. I will see where the name is written. So you see the name is written here, facing up. I will open this and place in the middle. And with my tweezers, I will press the antibiotic in the agar, and then I will close. Why do I need to press the antibiotic in the agar? To guarantee the agar will have contact with the antibiotic and the substance will go for the agar. If the antibiotic is not touching, you might have a false positive or a false negative. Wrong. So guarantee all the antibiotic discs is touching the surface of the agar. That's the very important part of this process. Okay. After the incubation time, that can take between 1 to 3 days, depending on the temperature, we will have the results. Okay, so I will show you the results and how to calculate. Your teacher will show you how to make a proper calculation for your necessity. Okay, so for the results, how are we gonna do to make the calculations? So as we can see here, we have one clear ring here and another clear ring here. Okay, there is a slightly shade in this one, and the other ones doesn't almost show anything. Okay, so we have three clear rings here. So I will show in this one that is best to see in the video how to make the calculation. So the first thing I need to do is find the center of my disc. I make a little spot, and then I will use a ruler and I will trace a line between the center of the spot and the edge of the spot. Then I will take a measurement. So this is 1.2 centimeters or 12 millimeters. Then I will do the same for this and the same for that. And then I will be able to calculate which one between these three have the best biotic effect, okay? When you're testing for different substances, the idea is exactly the same. Or you test to see if it works, or you test to see the best action, okay? Thank you for watching this video. If you like it, please give us a thumbs up. Please consider to subscribe, give us a comment, and see you next time.